So we were interested how those structural changes, which is the impact of those structural changes on the immunoadjuvant activity of those compounds. And this is the synthesis. I will just show it. It was a lot of, really a lot of work. This is these are last steps where we attached the oligosaccharides on this side here. And after that, we did global deprotection to deprotect all of those OH groups. And here are yields of those reactions. I will say that yields are quite decent if we take into account the complexity of those molecules. So we got all of those molecules and we test them for immunoadjuvant activity. Here are the results of antibody testing. Unfortunately, just our tetrasaccharide showed a tetrasaccharide with native structure showed the enhanced immunoadjuvant activity, which was significant immunoadjuvant activity, but other compounds were not active. So we didn't manage to really simplify this structure very much. So just from pentasaccharide to tetrasaccharide. But still, if you look at this graph, you can see that trisaccharide after 6 weeks shows some activity. And trisaccharide is much easier to synthesize than tetrasaccharide. And the results of other research group also shows that trisaccharide can be more active depending on what is found here on this position. So Livingstone's group prepared this compound shown here on the slide. They attached this chain with carboxylic acid at the end. And as you can see, this shown in red is activity of that trisaccharide, which is quite comparable to the activity of the QS21. Trisaccharide can be synthesized much more efficiently than tetrasaccharide in 24 versus 36 steps. And because of that, I decided to develop immunoadjuvants based on this trisaccharide in the future. And also in this very important paper is summarized the activity, structure activity relationship for QS21 derivatives. We can see on this structure what can we change on this structure and how that will affect the activity of immunoadjuvant activity of compounds. And they said that linear trisaccharide is required. So I decided to develop compounds with trisaccharide, not tetrasaccharide. After returning from my postdoc, I started independent research, and I have some new directions of that research, which I will show now. Those are based on molecular hybridization concept from medicinal chemistry. So if we have two compounds, both of them show some activity. Both of them, for example, show immunoadjuvant activity, but both of them have some problem in their activity. We can take parts of those molecules which are responsible for their activity, and we can put those parts by chemical synthesis. We can put those parts together in a new scaffold, and we can expect from this structure to have a higher activity and maybe to avoid some problems which were present for this compound 1 and compound 2. And my idea is to couple this trisaccharide with different acyl groups here with another saponin, which is other than saponin. For many other saponins, immunoadjuvant activity was known, but it is much less than for QS saponins. But many other saponins are much more abundant and much more easily available, and so much more cheaper. So I want to try to make compounds with this structure in order to test their immunoadjuvant activity. For doing that, I have to develop efficient synthesis of this trisaccharide. And I also succeeded in that. Before I synthesized this trisaccharide in this disaccharide in 42% yield, now I have 86%, and I also synthesized from this disaccharide trisaccharide 19 in a smaller number of steps, 12 steps instead of 16 steps in the synthesis which I did for my postdoc, and there is still room to reduce the total number of steps. Also, this method is much easier to carry out, is less moisture sensitive glycosylation. The main problem with glycosylation reaction is that it's extremely moisture sensitive. This method is not. So I developed the efficient synthesis of this trisaccharide, and I still have some room to improve it. And another idea is the molecular hybridization of two immunoadjuvant compounds. So we have here the saponin, which shows immunoadjuvant activity. And through this fucose derivative, I plan to couple it with another immunoadjuvant. Here on the Faculty of Science, for many years was done research on desmuromyl dipeptides and their derivatives, and I decided to put this compound together in one structure with saponin in order to test the activity of the prepared conjugate. Since both those derivatives themselves show some immunoadjuvant activity, I expect that this compound will show much higher immunoadjuvant activity, maybe due to a synergistic effect. And at the end, I will conclude. So despite being one of the most promising investigational immunoadjuvants, the clinical advancement of QS21 has been constrained due to its inherent drawbacks. So the main goal of research in this field is to simplify the structure in order to make it more synthetically accessible, but also more stable. And the approval of the QS21 in a vaccine for human use demonstrates the applicability of QS saponins. And in addition to vaccines against infectious diseases, QS21 has shown very promising results in clinical trials of anti-tumor vaccines, which gives a strong impetus to further research in this area. At the end, I want to thank to my parent institution where I work, to people with whom I work closely during last year, to my students who did their master thesis in this research area, and also to Professor Wang from United States, in his group I spent 1 year as a postdoc. He introduced me in this very challenging but interesting chemistry. And also many thanks to funding agencies, and thank you all for your kind attention.
Thank you so much, Dr. ŠKALAMERA, for so interesting and maybe so new information, especially for our student. And dear colleagues, do you have a question? I think we have some questions in the chat. The first question is, information. Dear Đani ŠKALAMERA, thank you for an interesting and meaningful presentation. And I have a question for you. Are there age restrictions for the use of supplement-based adjuvants? And the second question, can such adjuvants be used in vaccine for children?
I'm not aware that there are age restrictions for use those adjuvants, but the Shingrix vaccine is actually, I think that is for older people, because this is for herpes zoster disease. which is usually older people are affected by this by that disease. But also this adjuvant is developed because other adjuvants show some, let's say, side effects. So we want to avoid side effects, and this is a natural compound. So maybe it will not have side effects. It is approved for human use. It is tested in more than 100 clinical trials. So I think it should be quite safe for use.
And for children as well, I think so.
I think as well, yeah.
Yes, we have the next question. Is it possible to use adjuvants based on modified saponins for mucosal vaccines?
Probably, but they should be maybe more stable, because, for example, QS21 is prone to hydrolysis even in pure water, and if it is a little bit basic solution or just above 7 pH, it can hydrolyze, and then we have just TH2 immune response.
Mm-hmm.
So yeah, I'm not aware that it is developed some vaccine which use this some mucosal vaccine which uses QS21.
Thanks. And we have a next question from Andrei Saramot, Department of Molecular Immunology, Paladin Institute of Biochemistry. Thank you so much for fascinating talk. And the question is, what you tell about toxicity, bioavailability, compatibility with antigens, proteins, and polysaccharides of these modified and artificial adjuvants in host organism?
Yes, the toxicity can be a problem. The toxicity is dependent on the dose, and some of the saponins can show some toxicity, which we found so mice dies in those experiments. So the dose should be less, but those compounds are extremely immunoadjuvant active. So the dose which we use shouldn't be high enough to induce toxic effects. And compatibility with antigens, probably with any antigen will be compatible, because it induces a strong immune response, and actually I don't see a reason why it would not be compatible with antigens for subunit vaccines.
Thank you so much. Thanks again, Gianni, for so interesting presentation.
Thank you.